Hello, my name is Jeff Lin, and I'll be presenting you my poster on the effect of pathological phimosis on foreskin inflammation in pediatric patients. Pathological phimosis is the inability to retract the foreskin due to clinically apparent signs of inflammation, pain, skin irritation, scarring, local infections, and bleeding. Although the clinical symptoms of pathological phimosis are well documented, the underlying cause of the destructive inflammation remains largely unknown. Furthermore, surgical removal of the foreskin, otherwise known as circumcision, is currently the only treatment option available to treat patients suffering from pathological phimosis. However, why circumcision is an effective treatment option to pathological phimosis remains largely unknown. It has been shown that the elimination of the subperitoneal space under the foreskin through circumcision results in profound changes in the penile microbiome. Specifically, the reduction of strict anaerobic bacteria that have been associated with inflammation and increased infiltrating T cells. T cells, natural killer cells, dermal dendritic cells, and Langerhans cell populations have all been associated with skin-related disease pathologies such as atopic dermatitis and psoriasis. However, the immune cells involved in the development of pathological phimosis remains currently unknown. Therefore, studying them may provide clues to the underlying cellular cause of the clinically apparent inflammation and scarring associated with the disease. For this study, there are two objectives. The first objective is to develop immunofluorescent microscopy staining techniques to identify and quantify various immune cell types in human pediatric foreskin tissues. Then, the second objective is to determine the distinct immune cell populations involved in pathological phimosis. As for the methods, male pediatric patients seeking medical or elective circumcision from Children's Hospital at London Health Sciences Centre in London, Ontario, were invited to participate in this study. The inner and outer aspects of the excised foreskin samples were collected. Pathological phimosis and healthy controls were randomly selected for the analysis. And these were the results. Figure 1 shows immunofluorescence microscopy staining images of Langerhans, CD3, CD11C, and CD56 immune cell markers on human pediatric foreskin tissues. Panel A shows immune cells that were stained for CD11C and Langerhans, while panel B shows immune cells that stained for CD3 and CD56. CD11C stained for dermal dendritic cells, whereas Langerhans stained for Langerhans cells. CD3 stained for T cells, and CD56 stained for NK cells. In Figure 2, we see the overview of the pixel-based immune cell quantification methods that we have used and applied to the immunofluorescent microscopy staining images that we have taken. For each panel, on the left, we see the raw image. In the middle, we see the process image, where the background noise and autoimmunofluorescence has been removed. And on the right, we see the positive cell staining image, which shows what is positive cell staining in white and what is not in black. This sort of processing was done for each of the immune cell markers that we have used. Lastly, we compared the immune cell densities between the pathological phimosis group and our healthy controls. This is what is shown in Figures 3 and 4. We found that pathological phimosis was associated with elevated CD3 T cell densities, but only to the inner foreskin. No statistical differences were observed in outer foreskin CD3 T cell densities between the pathological phimosis group and the healthy controls. Furthermore, no statistical differences in NK cell, Langerhans cell, and dermal dendritic cell densities were observed between pathological phimosis group and the healthy controls. These findings suggest that the adaptive arm of the immune system, meaning T cells and not innate immune cells, are responsible for pathological phimosis. The observation that inflammation is localized to only the inner foreskin suggests that antigens specific to the subperitoneal space are potentially responsible in causing this inflammation. Future studies will investigate the antigen specificity of these T cells, other immune cells that may potentially be involved in disease progression, and potential association with pro-inflammatory bacteria. And that concludes the end of my presentation. Thank you for listening to my presentation on the effect of pathological phimosis on foreskin inflammation in pediatric patients. Before I leave off, we would like to thank all of the tissue donors along with all the men and women of London, Ontario for donating their time and efforts. I want to thank my supervisor, Dr. Jessica Prouger, our collaborators, and all the members of our lab for their support and guidance. Lastly, I want to thank the Department of Microbiology and Immunology at Western University.